Welcome dear students. Today we are going to discuss an important topic on metabolism and excretion of hormones. The main objectives of today's lecture are to give a brief idea about the hormones, to know why hormones are necessary, followed by a short reflection on the action of hormones, besides knowing about the effects due to hormones, and finally to understand the mechanism and secretion of hormones in detail. Dear students, let us first talk about what hormone is. A hormone is a chemical messenger that coordinates the activities of different cells in a multicellular organism. The term hormone was introduced by Starling in 1905. The first hormone to be discovered was secretin. It was discovered by two English physiologists, William N. Bayliss and Ernest H. Starling in 1903. Secretin is a hormone produced by the duodenum to stimulate the flow of pancreatic juice. Hormone is a chemical substance that is synthesized by a particular endocrine gland, which then enters into the blood circulation to be carried to distantly located target cells or organs which have specific receptors that bind to it on the surface. They serve to coordinate the metabolic activities of several tissues or organs. They accelerate or inhibit specific physiological processes, and they do not initiate a reaction but can influence its rate of action. Dear students, moving ahead, we will now know why hormones are necessary. Hormones are involved in maintaining homeostasis, that is the consistency of the internal environment that is maintained for the benefit of whole organism. The concept of homeostasis was first recognized by Claude Bernard in the 19th century, who noted that the internal environment, that is the fluid bathing cells, had to be regulated independently to the external environment. Being able to regulate and maintain the internal environment gives an animal a freedom from change in the external environment, allowing it to live in a changing or even harsh environments. However, there are metabolic costs associated with maintaining homeostasis by negative and positive feedback mechanisms. Firstly, negative feedback mechanism. When the secretion of a hormone is inhibited by an effect produced in the target cell, for example, follicle stimulating hormone that stimulates the secretion of estrogen from the ovary is suppressed when estrogen levels reach a particular concentration as shown in the given figure. Secondly, the positive feedback mechanism. Secretion of certain hormones is stimulated by the effect it produces. For example, oxytocin from the posterior pituitary gland enhances uterine contractions during parturition. This causes the baby to descend to the cervix, further stretching the cervix, which in turn stimulates the release of oxytocin as is shown in the figure. Dear students, until now you might be well aware about the term hormone and why it is necessary for us. Now we will discuss briefly about the action of the hormones. Hormones are very specific and only exert their effects on target cells which possess the particular protein receptors that recognize the very hormone. Non-target cells lack such receptors and do not respond to the circulating hormone. Hormones alter cell activity by either stimulating or inhibiting a distinctive cellular response to the target cell. Target cell responses to hormone stimulation may involve change in membrane permeability, enzyme synthesis, activation or inhibition, secretory activity, gene activation, and mitosis. Once attached to a receptor, hormone may exert its effect in numerous ways. Dear students, by now you have known how receptors are very important for hormone action. Moving to next objective, that is knowing about the effects due to hormones. Hormones cause change in cellular metabolism. They do not directly cause change in gene structure, but can activate genes to influence gene expression and ultimately protein synthesis. Hormones can alter catalytic rates of enzymes by mechanisms such as the phosphorylation and dephosphorylation of proteins. Hormones can also alter membrane permeability to affect transport, processes, and ion movements, alter muscle contraction, exocrine secretion, and water permeability. All these general mechanisms cause a variety of effects in an organism. Hormones can cause morphological change as well, such as the differences in body shape between the male and the female. Hormones act as mitogens to accelerate cell division or alter gene expression to trigger differentiation of cells, stimulate the overall rate of protein synthesis or the synthesis of specific proteins. Hormones are also involved in stimulating smooth muscle contraction. For example, oxytocin stimulates contraction of the myoepithelium in the mammary glands for milk ejection. These also affect exocrine secretions. For example, secretin, a peptide hormone from intestinal mucosa that stimulates pancreatic secretions. Hormones also control endocrine secretions and a number of trophic hormones from the anterior pituitary. These can also stimulate or inhibit hormone secretion from target organs. Hormones regulate ion movements across membranes and control permeability to water. For example, antidiuretic hormone or ADH or vasopressin increases water reabsorption by the kidney, and ultimately hormones have a dramatic effect on behavior such as sex-related behavioral characteristics in males and females, maternal behavior, nesting activity, and broodiness. To sum up, now we will know about the metabolism and secretion of hormones. Dear students, most of the hormones in the body are polypeptides and proteins. These hormones range in size from small peptides with as few as 3 amino acids, for example, thyrotropin releasing hormone or TRH, to proteins with almost 200 amino acids, for example, growth hormone and prolactin. Polypeptides with 100 or more amino acids are called proteins, and those with less than 100 amino acids are referred to as peptides. Protein and peptide hormones are synthesized on rough endoplasmic reticulum of different endocrine cells. They are usually synthesized first as larger proteins that are not biologically active, as preprohormones, and are cleaved to form smaller prohormones in the endoplasmic reticulum. These are then transferred to the Golgi apparatus for packaging into secretory vesicles. In this process, enzymes in the vesicles cleave the prohormones to produce smaller biologically active hormones and inactive fragments. The vesicles are stored within the cytoplasm, and many are bound to the cell membrane until their secretion is needed for the action. Secretion of the hormones as well as the inactive fragments occurs when the secretory vesicles fuse with the cell membrane and granular contents are extruded into the interstitial fluid or directly into the blood circulation by the process of exocytosis. In many cases, the stimulus for exocytosis is an increase in the cytosolic calcium concentration caused by depolarization of the plasma membrane. In other instances, stimulation of an endocrine cell surface receptor causes increased cyclic adenosine monophosphate or CAMP and subsequently activation of protein kinases that initiate secretion of the hormone. The peptide hormones are water soluble, allowing them to enter the circulatory system easily, where they are carried to their target tissues. Since steroid hormones are usually synthesized from cholesterol and are not stored, the chemical structure of steroid hormones is similar to that of cholesterol, and in most instances they are synthesized from cholesterol itself. They are lipid soluble and consist of 3 cyclohexyl rings and 1 cyclopentyl ring combined into a single structure, as is shown in the figure. Although there is usually very little hormone storage in steroid producing endocrine cells, large stores of cholesterol esters in cytoplasm vacuoles can be rapidly mobilized for steroid synthesis after a stimulus. Much of the cholesterol in steroid producing cells comes from the plasma, but there is also a de novo synthesis of cholesterol in steroid producing cells. Because the steroids are highly lipid soluble, once they are synthesized, they simply diffuse across the cell membrane and into the interstitial fluid and then the bloodstream. Whereas amine hormones are derived from tyrosine. The two groups of hormones derived from tyrosine, the thyroid and the adrenal medullary hormones, are formed by the actions of enzymes in the cytoplasmic compartments of the glandular cells. The thyroid hormones are synthesized and stored in the thyroid gland and incorporated into macromolecules of the protein thyroglobulin, which is stored in large follicles within the thyroid gland. Hormones secretion occurs when the amines are split from thyroglobulin and the free hormones are then released into the bloodstream. After entering the blood, most of the thyroid hormones combine with plasma proteins, especially thyroxine binding globulin, which slowly releases the hormones to the target tissues. Epinephrine and norepinephrine are formed in the adrenal medulla, which normally secretes about 4 times more epinephrine than norepinephrine. Catecholamines are taken up into preformed vesicles and stored until they are secreted. Similar to the protein hormones stored in the secretory granules, catecholamines are also released from adrenal medullary cells by the process of exocytosis. Once the catecholamines enter the circulation, they can exit in the plasma in free form or in conjugation with other substances. Each of the different hormones has its own characteristic onset and duration of action, each tailored to perform its specific control function. For example, epinephrine and norepinephrine are secreted within seconds after the gland is stimulated, and they may develop full action within another few seconds to minutes. The actions of other hormones such as thyroxine or growth hormone may require months for their full effect. The concentrations of hormones required to control most metabolic and endocrine functions are incredibly small. Similarly, the rates of secretion of the various hormones are extremely small, usually measured in micrograms or milligrams per day. There are highly specialized mechanisms available in the target tissues that allow even these minute quantities of hormones to exert powerful control over the physiological systems. Steroid hormones are not stored but released immediately to diffuse out of the cell. Protein and peptide hormones are stored in granules within the gland and are released in response to various stimuli. The other tropic hormones like FSH and LH, that is follicle stimulating hormone and luteinizing hormone, stimulate the synthesis and release of gonadal steroids, while adenocorticotropic hormone or ACTH stimulates the synthesis and release of adrenal steroids. Hormones can also be released in response to nervous stimuli from environmental cues such as light, smell, sound, and temperature. This neuroendocrine transduction illustrates the integration of the nervous and endocrine system. Hormones are also released in response to a number of metabolites. For example, intracellular glucose levels control glucagon and insulin secretion, amino acids stimulate somatotropin release and increased uptake of amino acids, while extracellular calcium ions regulates parathyroid hormone and calcitonin secreting cells. These effects are all examples of stimulus response coupling. Dear students, in conclusion, hormones must be metabolized rapidly and removed so that feedback mechanisms can operate and hormones can regulate cellular functions. Removal or inactivation of hormones follow exponential decay kinetics as is shown in the figure. The half-life of the hormones in the circulation is a measure of the longevity of hormone action. Many synthetic hormones and hormone analogs are designed to have a longer half-life and thus be effective for longer periods of time than natural hormones. Peptide hormones are degraded by peptidases such as the cathepsin in lysosomes, which split the peptide bonds in the molecule. Exopeptidases degrade protein from the carboxyl terminal end or the amino terminal end. Endopeptidases such as trypsin and chymotrypsin degrade the protein at internal sites with some specificity. Trypsin hydrolyzes peptide bonds where the carboxyl group is from lysine or arginine, while for chymotrypsin, the carboxyl group in the peptide bond comes from phenylalanine, tryptophan, or tyrosine. Deamination or reduction of disulfide bonds, for example, insulin, can also inactivate proteins. This occurs in kidneys, liver, and in target cell lysosomes. Steroid hormones are bound to protein carriers in blood, such as serum, albumin, or steroid binding globulin, which are necessary since steroids are lipophilic. Binding to protein carriers also increases the half-life of steroids. Physiologically, only 5 to 10% of the hormone is present in the free or unbound form. Steroids are degraded by a two-phase process in the liver and in the kidneys. This process inactivates the steroids and makes them more water soluble for excretion. In phase 1, enzymes such as cytochrome P450 or CYP add functional groups such as hydroxyl groups. These metabolites are then conjugated to glucuronic acid or sulfates by transferase enzymes as is shown in the figure. And finally, these more water-soluble metabolites are excreted by the kidney in urine or by the liver in the bile salts. Dear students, this was all about today's lecture. Hope you have understood it well. Thank you and goodbye.